biology students, what we're going to be talking about today is transcription and translation. Actually, we're going to be specifically talking about RNA and transcription. We'll leave translation to another time. So one of the things we're going to discuss here is we're going to be discussing the genetic code. If you remember, we did an activity called the M&M Shuffle, where we were looking at how a student might be able to eat M&Ms in a certain order. So for this case, if I would eat, of the 4 colors of M&Ms that I had, if I ate them one at a time, how many different combinations could I have? Well, in that case, I could only have 4 different combinations. Okay. Now, what would happen if I would put 2 M&Ms together and having the order be, specific? It can be variable in some ways, but very specific. If I eat a blue one, I could eat another blue one. Well, if I eat a blue and a red one, that's one order. Specifically, I could eat a red and then a blue. That'd be a completely different order. So those are things that can occur. So if I were to eat of 4 different colors, if I had 2 different M&Ms at one point, then that would mean that I would have 2 to the 4th possibilities, or 16 different possibilities that are possible. Now, in the other sense, if I would do 3 different possibilities, then this would actually show me that not only could I have 4 different lead colors, I'd have 4 different secondary colors on those lead colors, and I'd have 4 different colors on the secondary colors. So it actually turns out to be 64 different combinations of those M&Ms. Now, why do I talk to you about that? Why do we start with that exploration? Well, it turns out for genes, to make proteins, or to carry out processes in the cell relies on DNA to hold on to genetic information, and specific genetic information that will actually put forward information to create proteins which are essential to life, things like enzymes which make chemical reactions go faster, things like structural proteins like muscles and tendons and ligaments. Those things are held together. And other specific processes about when to do certain actions and when not to do certain actions. Those are all controlled by genes. So all that's very, very essential. So for DNA, in the sequence, we have to be able to code within that DNA sequence for enough amino acids to have all the proteins that are essential to us. So in this slide that you see here, you see that we're looking at the synthesis of protein is actually is going to be a process we're going to call transcription and translation. But here's the main part of this. We're going to have 20 different amino acids that we're going to be dealing with. So those 20 different amino acids are not covered if, say, adenine, thymine, cytosine, and guanine are taken in one at a time. If we look at a gene as being only coded for one nitrogenous base, like adenine, well, there'd only be, really, of the 4 nitrogenous bases, there'd only be 4 amino acids that we could code for. So that wouldn't work. Well, if we took 2 nitrogenous bases together and had that be a code, so say we take adenine and thymine together and have that be a code for an amino acid that we might need, then we would only end up with 16 possible amino acids that we could code for, which is some, but not all of the amino acids we would need. So in the end, we actually take amino acids in what's called a codon. So this is one of the main terms we're going to be talking about here with our objective sheets here is the idea of a codon. What is a codon? Well, a codon is basically 3 nitrogenous bases that are paired together, and it codes for 64 different amino acids, and actually specifically 61 amino acids are actually coded for, 3 of which are actually just stop codons. So, like I said, the codon here is just 3 nitrogenous bases. Here we have a code which would be adenine, 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 and that specific codon will actually code for the amino acid right here shown. So, again, the idea that there are 64 different possible codons based on our nitrogenous bases, and there are 61 codons that will actually signal for amino acids. 3 of the codons actually will code for what's called a stop protein, which when we get to the idea of translation, that will make a little bit more sense of why do you need to stop. But for the most part, it will tell the ribosome when to stop signaling for tRNA. We'll talk about the RNAs here in just a second. So that's kind of the basics of why do we have, what was that M&M shuffle about? But it's the idea that we need to have genes, and these genes are coded for by nitrogenous bases in a 3 nitrogenous base sequence called a codon. Those codons specifically look at things like, or code for amino acids. And amino acids are essential for creating proteins and enzymes that are necessary for life. Okay, so that's the basic premise of where we're at, the idea of codons and genes. Okay, so now, and this is a genetic code sequence that we can kind of, not a sequence, but a structure, a diagram that actually shows us the nitrogenous base codon sequences and what amino acid they actually code for. Okay. Now, we are going to get into transcription. Transcription itself is a process of taking the DNA sequence and creating a RNA sequence that will eventually leave the nucleus and enter the cytoplasm. Okay, so one of the first things we need to discuss is RNA structure. RNA structure is different than DNA. I have a nice little picture over here for those of you that are going to be paying attention long enough to look at it. There are 3 main reasons or things that are different between RNA and DNA. First of all, RNA is a single strand. If you look at the picture over here, DNA on my right side has 2 different strands and a double helix. Okay, RNA is a single helix. It's just one side, so it's not meant to be held together as far as everlasting. It will eventually be destroyed when its usefulness is done. So it really has only just one side, and eventually that side will be read by the ribosome to create protein. RNA has the sugar ribose. Okay, so if you look along the DNA strand, you'll notice that in a nucleotide you would have a phosphate group, then a sugar, which is deoxyribose in DNA, and then a nitrogenous base. In RNA, we actually have a nucleotide, which is phosphate group, ribose, and then nitrogenous base. So the sugar is a little bit different, thus the difference in the name. And then finally, RNA has the nitrogenous base of uracil instead of thymine. So when we're putting together an RNA strand, we actually have uracil, and I am highlighting it right now with the laser pointer here. So the uracil is actually the substitute in there for thymine. So wherever, when we're base pairing from a DNA into an mRNA strand, where we're going to talk about in the next screen, we're actually going to put a U where we would actually put T. Okay, so here's examples of the types of RNA. There are 3 types of RNA. The first is a messenger RNA. Messenger RNA's specific goal, and you'll need to write this down, specific goal is to take the DNA sequence that's been copied and bring it to the ribosome. So it's a messenger RNA. So messenger RNA is created from the DNA. So DNA creates this mRNA strand. This mRNA strand will actually leave the nucleus and go to the cytoplasm. The next one is called transfer RNA, or tRNA. Transfer RNA is specifically responsible for bringing amino acids to the ribosome in the cytoplasm. So this is more a part of translation. This is more a part of translation. And ribosomal RNA is just kind of the housing, the ribosome itself, where tRNA and mRNA will meet up. Okay, so the final couple things we'll talk about here is specific to transcription. Transcription is the first process in creating a protein. So again, what's the purpose of all this, Mr. Osmith? But the idea that we're shooting for here is we've talked about DNA. We've talked about how to take DNA and create and how it replicates itself during S phase of interphase. And what we're trying to do now is show you how those genes that we talked about in genetics are actually expressed and what they're meant to do. So these genes are actually coding for proteins. So this is kind of the next step in the maturation of our ideas of genetic information. So in this case, we're going to actually discuss the process in 2 steps. The first step here is transcription. So an enzyme will unzip. Okay, so it will unzip DNA, and then it will, of course, uncoil. And inside the nucleus, we're going to create an mRNA strand using these free RNA that are just kind of hanging around the nucleus. And in those cases, we're going to create this mRNA strand. And that's going to go ahead and break away from the DNA once it's done creating the specific gene that we're looking for. The mRNA will then leave the nucleus and will enter the cytoplasm. All right, so here's an example of what we're talking about. So DNA itself will actually unzip, and then using these free RNA nucleotides, we're actually going to base pair. Now, here's the thing about base pairing, and this will be the thing that you're going to really want to make sure you get done on the worksheet. When we're base pairing, this is the DNA strand. This is the mRNA strand that we're going to create based on this. This messenger RNA is going to actually leave the nucleus and go to the ribosome. But notice as we base pair, cytosine is going to pair with guanine. Adenine is going to pair not with thymine down here, but with uracil. Okay, so uracil is the substitute in for thymine on RNA strands. All RNA strands will not have thymine. They will have uracil. Okay, so adenine will pair with uracil, adenine with uracil. But then as we come over here with thymine on the DNA strand, thymine's on the DNA strand still. That's why it's still there. It will pair up with adenine on the RNA strand. So adenine's still part of it. So A pairs with U on RNA, and T will still pair with A. And then we can finish out here just normally. Of course, the U is down here at the end because it's being paired with A on the mRNA strand. All right. So a couple of review questions for you. What organelle is responsible for making proteins? That's called a ribosome. Where a ribosome is found in the cell, in the cytoplasm. If the DNA is found in the nucleus and has a genetic code, then how does it reach the ribosomes in the cytoplasm to make proteins? The specific answer is RNA. Okay, so we will be talking about translation tomorrow. Thank you very much.